All right, I'd like to get started by introducing our speaker. Dr. Karla Satchell is a professor of microbiology immunology at Northwestern University Feinberg School of Medicine. Dr. Satchell also leads the Center for Structural Genomics of Infectious Diseases, a consortium of 9 labs at 8 schools collaborating on an effort to examine the atomic structure of COVID-19. Let's welcome Dr. Karla Satchell.
Oh, it's great to be here today. Thank you. So I'll go ahead and share my screen and then we can start. Well, I want to thank everyone for inviting me here to talk today. It's really great to talk to the C2ST audience. I'm going to talk today about structural biology for drug discovery and really how to tackle COVID-19 from a very basic standpoint. So, where should we get started if we wanted to make a new drug starting today? So I wanted to just mention that I'm the co-director for the Center for Structural Genomics of Infectious Diseases, which is a large NIAID-funded center that's headquartered at Northwestern University, Feinberg School of Medicine, and we've been in existence since 2007. But we're really a very large consortium, international, with 8 institutions, including UC Riverside, University of Chicago, and Argonne National Labs, University of Virginia, Purdue University, UT Southwestern, University of Calgary, and WashU. And so I'm really, what I'm going to present today is on behalf of the entire consortium. So we've actually been working since all the way since January 10th on the coronavirus. How we got involved in this so early is that because we are funded by NIAID and we're what's called the Biological Resource Center for them, we're actually part of the existing infrastructure that NIAID has in place for in order so that they have trained staff, equipment, and technologies in place should there be an emerging infectious disease. And so we and as well as our cohorts in genomics and bioinformatics and preclinical testing were activated as soon as it became clear that this was going to be a significant emerging infectious disease event. The virus that has come out is very close to SARS-CoV-2. Both of these viruses are small particle viruses with these spikes that extend out, and they actually originally, when viewed by electron microscopy, were thought to look like a crown, and therefore coronaviruses were referred to as, that's where the name corona comes from, because corona is the Latin word for crown. The particle itself is 15 to 200 nanometers in diameter and is made up of only 4 proteins. And those proteins are organized as shown in this graphic. And we see that the viral particle is circular and that's constructed of 2 proteins, which is the membrane protein, or the M protein, and the envelope pentamer, which is the E protein. And this is embedded in membrane that actually buds out from our human cells as the virus gets made. Extending out across that membrane is this very important protein called the spike glycoprotein trimer, and that's shown here in yellow, and I'll show this in more detail in just a moment. Inside that viral particle, we have the nucleocapsid protein, which is a small protein that wraps the genomic single-stranded RNA that is the genetic material of the virus. Because that RNA has to be wrapped in this protein, it is the most abundant protein of the entire viral particle. But the entire virus is composed of just these 4 viral proteins and the RNA. This terrible small little virus then goes on to cause devastation when it interacts in our lungs. Here we see a very early x-rays of a patient in China. We see a normal lung, really great lungs, actually appear black because that's empty airspace in an x-ray. But when you get congestion and your lungs begin to fill with fluid, you see this white begin to appear, and that gets deeper and denser as time goes by. And here we see a patient 4 days later that has this very deep congestion, deep pneumonia, and leads into acute respiratory syndrome that then can be lethal. So what can we do to treat this terrible disease? And we want to think about new drug discovery in multiple different levels. First off, we can repurpose current drugs. And this is something that can be very fast. These are drugs that are in the clinics, they're FDA approved, and the safety of these drugs is already well documented. things that we can think about in terms of purposing new drugs is things that treat inflammation. So that pneumonia actually is a lot of inflammation involved there. And so there's lots of interest in looking at drugs that are already in place to control inflammation and whether they can be used in critical patients to help them improve, to avoid death. But we can also think of antiviral therapies. And these are drugs, those small molecules that will be directed against the actual viral particle. And these could be used much earlier during infection, during that acute viral infection phase, before this deep pneumonia and immunological syndrome sets in. And this is where our center has really been thinking about potential therapeutics. Here as well, we can think about repurposing current drugs. There are many drugs that have been developed for treatment, for example, of HCV or HIV, and there's lots of interest in testing if these antivirals can be used to treat this particular virus, and that's involved in ongoing research. We can also think about antibodies. Antibodies can be identified in human antibodies from patients who have been exposed to the disease. As we're now 3 months into this disease syndrome, there are hundreds of thousands of survivors in the world that we can actually probe their immune response to say, what could we do to look at antibodies as potential therapies to block the viral infection? And this can be medium fast. The major issue right now is you needed a pool of patients, which we now have, but then you need the development of that technology, which is ongoing now. You can also think about rebooting SARS research. SARS occurred in 2003. It's a virus quite close to this particular virus. And there was extensive research that went on from 2003 really until around 2010, picking up again around 2012 with the advent of MERS, which is sort of a distant cousin of this virus. Because that research was in many cases halted or was not particularly advanced, this is going to be a medium slow response. And that's because the work still needs to go through advanced stages of laboratory-based development as well as clinical trials. And then finally, we can just have de novo drug discovery. This is by far going to be the slowest, but this will lead potentially to the most specific antivirals because it will be designed specifically against this particular virus, but the timeline is likely to be very long. But we shouldn't shrug off this as a reason to not do it, because if we hadn't stopped SARS drug development back in around 2010, we would have a drug because this virus is so close that things effective against SARS are likely to be quite effective also against this virus. So if you think about the viral life cycle and where we could actually think about developing drugs, this is a very cartoony schematic that allows us to very simply look at the various stages. When a virus enters into your lung, it's going to attach to that cell. It has an entry mechanism that penetrates across the membrane to then enter the cell. That viral particle is then uncoated and that nucleocapsid is spilled out and that nucleocapsid falls off, and now you have a naked mRNA present in the cytosol of the cell. That is then, there's biosynthesis that goes on, that mRNA is read, new mRNAs are made, and the proteins required for synthesis as well as assembly of that particle are made, and we see the reproduction of new viruses in the cell that then burst out of that cell and are then free to go infect the neighboring cell in the lung. And so this is the general life cycle. So when we think about drug discovery, we think about multiple levels that are druggable. We can use these antibodies that could then bind that viral particle, which would then block that entry stage. So this can be a very productive way to halt viral progression. Penetration is a place where you can have druggable events by targeting those events that are required for the host cell. And this is where chloroquine or hydroxychloroquine has its effects. One of the issues with those two drugs is while they're very effective for cells in culture, it's not clear that they're particularly effective when you get into the lung space. And so this is why it's very important that we have ongoing drug trials to test if chloroquine is going to be effective. And then finally, for biosynthesis, this stage down here, you have many different machines that are built by the virus in order for this assembly process to happen. And each of those machines is an enzyme. Enzymes make excellent drug targets. And so knowledge of those can actually help us develop a new drug. So if we just look at a more detailed life cycle, we get into a lot more events here. This is much more specific to the coronavirus, but I'll just use this as a guideline to take you through some of the places that are being looked at for drug development. And that first stage is going to be right up here on top, where we see that this spike protein on the surface of the virus engages the surface receptor. That surface receptor is angiotensin converting enzyme 2, better known as ACE2. If we look at the structure of the spike trimer, we see that that trimer actually is present on the surface of the virus, which I showed in that earlier cartoon, and it extends out as a trimer of proteins. It actually has two domains, the S1 domain, which is more like the head, and the S2 domain, which would be more like the stalk. And within that S1 domain, we have a small region of that protein that directly engages that receptor. And the receptor in the middle cartoon shows that engagement occurring. But there's steric clashes, which means all three do not bind the receptor, just one of those. And so there's movement of that spike that allows to resolve those steric clashes so that you get very tight engagement of that receptor. There is a processing event that occurs. That processing is mediated by proteases on the surface of the cell. You now get maturation of that viral particle through conformation change, and then you get membrane fusion. And so the membrane of the viral particle fuses with the human cell, and that's how we see that viral particle engage and enter into the eukaryotic cell. This has been looked at in detail, the structure biology. This is a cryo-EM structure. This was not solved by my consortium or our consortium. This was solved by our sister group, which is the Seattle Structural Genomics Center for Infectious Diseases. They are also the NIAID-funded resource center, and we partner with them on many different studies. And here we see the detailed structure of that trimer in both that closed format, which is going to undergo those steric clashes, but now also in that open format where we see that conformational change, we see an extension of that receptor binding and binding domain that then engages the ACE2 receptor. If we look at the ability of this to bind versus SARS-CoV-1, which also binds the ACE2 receptor, we see that that KD, the number here, 1.2, is slightly lower than the number here of 5.0 nanomolar. So that means it binds slightly tighter. But if we look at the kinetics of that, you see that the next number here is the K on, which is how fast it engages. And that actually is just exactly the same. What's different is that when it binds, it stays bound longer. And that's what we call the K off. And so it engages and binds longer than the SARS-CoV-1. And so it's a slightly better binder because it engages longer. Another really interesting aspect about the spike trimer that's different than what we see for SARS-CoV-1 is that the sequence of it is these 4 residues larger. So I mentioned that there's 2 domains, an S1 domain and an S2 domain. And right at that point of cleavage, there's this 4 extra residues. And that converted the cleavage site to a furin cleavage site. And furin is a protease that's present on the surface of human cells. And so this means that the trimer in its maturation is now closely tied to the human cell. And it's thought that the gain of this particular sequence assisted in movement of this from a bat into human cells and had to do with moving from a zoonotic disease to a human disease. We see that that cleavage does occur if we take the viral particle and look at the S protein in it. You see in SARS-CoV-1 in the first lane that protein is a full-length protein, but in SARS-CoV-2, it's actually split already into the S1 and S2 portions. Although in the 3-dimensional structure, these hold together. A mutant that takes away those 4 extra residues, now we don't see that. But surprisingly, when they went in and did in vitro studies, they found in these two middle bars that there was not a particular difference in the ability of the virus to enter. And even when they made that mutation, at least in this study, they saw slight enhancement. So it leaves it unclear whether this leads to enhanced viral entry, or this maybe had mostly to do with jumping species. But we also don't know what's the difference when we get into our lung cells. Even a slight variation, slight enhancement in vivo can lead to a very major difference in terms of pathogenesis. So where's the next step going for studies of spike? And that's now, spike, of course, is on the surface of the virus. This is an excellent place for antibodies to be directed and is really a major antigen for our own systems. And so there's great interest to identify antibodies in human sera that specifically engage spike and neutralize the ability of spike to engage that ACE2 receptor. There's a big study that came out just a week ago, or just this week, that showed that there's a antibody that actually engages an antigen that is shared between SARS-CoV-1 and SARS-CoV-2, and this could be more broadly applicable across the coronavirus, and thought to be a very major advance in neutralizing antibody detection. So at this point, I'm going to leave behind the receptor and we'll move deeper into the viral life cycle. That virus, as I mentioned, enters into the cell, and now we see the mRNA being discharged into the cytosol. There, our host ribosomes recognize that RNA and begin to translate that RNA into proteins. And that translation event leads to two very large proteins, ORF-A and ORF-AB. And so I'm going to blow this up and look at it larger here in this next slide, where we see ORF-A1 is the first open reading frame. Due to a slip strand translation, that can actually be translated in some cases into a much larger protein that is ORF1A plus ORF1B. This large protein is then proteolyzed by two proteins that come along in this translational product. The first one is PL-PRO, for papain-like protease. So that papain-like protease cleaves this polyprotein in three positions. First off, it cleaves itself off between this position NSP2 and itself, and also cleaves itself from NSP4, and also cleaves here NSP1. So this now releases three proteins available for viral development. The next protein that's important is NSP5. That's the fifth protein. It cleaves itself away from NSP4 and then also cleaves all the way downstream down this polyprotein. Ultimately, we see release of 16 different proteins for viral development. And this is the first important stage in the virus. So proteolysis of ORF-AB and inhibition of these two proteases, 3CL-PRO and PL-PRO, is considered to be the most druggable event in the coronavirus life cycle. If we look across all antiviral therapies, large numbers of those are anti-proteases, because all viruses need to go through proteolysis as part of their development. So this is one of the places we're looking at repurposing old drugs, but also where we're looking at development of new drugs. So some of the developments that are going on with regards to development of new drugs include this work that was done at the Diamond Light Source in the UK. And what they did was what's called a fragment screen. And so they took the they took the M-Pro, or this 3CL protease, and did crystallization of that 1,300 times, bound to 1,300 different, attempted to get it down to 1,300 different ligands. Those release structures, they identified for a total 66 different active fragments that bound to this protease, 44 of which were covalently bound. And the idea here is that they identified all the little pieces that could be assembled to make a full-size drug. So if you know those pieces in the pockets they bind to, chemists can look at that and begin to assemble that in thoughtful ways to develop new drugs that would inhibit this protease. If you notice the dates here, you'll see that these were released on March 17th as well as on March 24th. How is it then that actually we used all those pieces already to release a structure from our center by March 25th? Here we see against that 3CL protease bound to a really beautiful drug that we call compound 77. And when we looked at the fragment screening, we noticed that many of the fragments that were identified by the Diamond Light Source study are actually present in this drug. And one of the reasons why we were so successful to get to a drug candidate so quickly is that we had been studying within the center, we've been studying coronaviruses for really more than the past 2 years as a major drug discovery component. And the idea of how we got it, of how this was involved, is this was the genesis of Andy Messakar, who's a researcher in our center at Purdue University. And his work goes all the way back to the SARS epidemic. And his idea was that if SARS came out in 2003 and MERS came out in 2011 and 2012, that it was just a matter of time before a new coronavirus would emerge. And the ideal drug wouldn't treat SARS or MERS, but would actually be able to treat the proteases all around the phylogenetic tree of known coronaviruses. And so he was developing drugs that would target all across the alpha, beta, and gamma coronavirus families. And from that screen is where compound 77, which is our lead compound, came from. And so this work is ongoing. They're looking at modifications to this lead compound, as well as looking to move this into viral testing, as well as into other preclinical trials. The other protease is also a very excellent drug target. That's the papain-like protease shown here. The center recently solved the structure of this one. It was released on April 1st. And this is now going to follow a very similar pipeline of fragment screening as well as ligand screening, and then to look for a new drug. Again, this has also been under study in our phylogenetic pathway, and so we have thought that we have lead compounds that could be looked at quite readily, although this was not as advanced as the other protease. And so finally, the proteases are great places for repurposing FDA-approved drugs. Under a study that we're doing in collaboration with NIAID, we're testing FDA-approved protease inhibitors and other compounds for inhibition of activity of these proteases. As part of the ligand screening, we had to develop nice high-throughput studies, and this was taken advantage of, and this is in collaboration with NIAID, who has us doing this testing. And we hope to have results of that to share sometime soon. So we've looked at the translation step, making PP1A and 1AB. We now know that proteolysis is important. And now what do we make with all these proteolytic fragments? The main piece that needs to be made is the replicase. And the replicase takes that mRNA for transcription, but also for replication to make the viral DNA. And that can be shown here in terms of what that transcriptome looks like. So the RNA here needs to be made into additional transcripts to make the spike protein. Here we see the E and the M protein, that nucleocapsid protein, as well as some other small proteins who have less defined functions. So that replicase is made by a number of different proteins that form a complex. The cryo-EM structure of that complex was solved by a group in China, Jihe Rao, and in Beijing. And his group has been very active looking at structures of SARS and MERS over the last 5 years. And here we see this beautiful cryo-EM structure that they deposited on March 17th. And that's composed of 2 proteins released from the polyprotein NSP10 and NSP7 and NSP8. And we see that this complex, they say, looks like like a fist, where we have the palm and the fingers and the thumb with a hole in the middle of it, and 2 molecules of NSP8 and 1 molecule of NSP7 bind into this protein. And this is the replicase that takes that mRNA and turns it into into DNA from which new transcripts can be made. Into this complex, it's thought that there are 2 additional proteins. This NSP9 protein, this small protein here, we solved the crystal structure of in the CSGID at 2.95 angstroms. It's thought that this must bind into this replicase at some point, but no one has ever visualized that. Another protein is part of this replication machinery is NSP15, which is an endonuclease. This was the first structures of SARS-CoV-2 that was solved by our center, and both of these structures were solved by Andrzej Wikimiak's group, the University of Chicago, and his laboratory is housed directly at Argonne National Labs. And how these two structures fit in is not yet known, but NSP15 in particular is a really nice drug target. Drugs were developed for this in terms of treatment of SARS, and they're trying to reboot that research now in the context of SARS-CoV-2. And the other very important aspect of looking at this complex really has to do with remdesivir. Remdesivir is considered to be one of the biggest, most promising repurposing of drugs for treatment of SARS-CoV-2 and is actively in clinical trials all throughout the world, including at Northwestern and other sites in Chicago. The remdesivir, I show the chemistry of this here, and it was published in a paper that was just came out actually in February of 2020, although it must have been in the works much before that, that this compound actually inhibits the complex that you see here, and it inhibits it directly with regards to treatment of MERS, and it's been shown to have efficacy in patients for treatment of MERS. And so this is really the most promising drug. It's not an outstanding drug, but it's a very good drug in preclinical tests. And it would be really great if we could get this drug bound into this complex and see what that looks like structurally, so we would know maybe a slight modification would change this from being a very good drug to an amazing drug. And that is ongoing research. So if we come back to our transcriptomes, we now have made these transcripts as the virus goes through its life cycle. The next thing that has to happen for efficient translation is that these transcripts actually need to be capped. So they are capped by the normal the normal host cap actually adds that first cap on. But when that cap is added to the end of the mRNA, it needs to undergo a methylation. And that N7 methyltransferase is actually brought in with the virus, and that's a protein called NSP14. To be highly active, it needs to be bound to another one of these proteins, NSP10. The second modification that has to happen is you need to have a second methyl added by a 2O methyltransferase, which adds a methyl group at this position shown here for the Cap1 construct. That's actually also really important because most viral RNAs don't have that cap. And so our host cells are attuned to RNAs that aren't supposed to be there. But when you add a methyl group there, it makes it look more like a host methylation, and that makes it hide from our immune system. SARS-CoV-2 brings in a specific methyltransferase to add a methyl group at that position, and that's encoded by NSP10 and NSP16. So if we look at the structure biology progress here, the structure of NSP10 and NSP14 has not yet been solved for SARS-CoV-2, although there's active work going on. But the structure is expected to be 95% identical to the structure that was already solved for SARS, which was done again by Zhihao Rao's lab in Beijing. And we see here the NSP10 shown here in red. This is the activator protein. And then you also see the NSP14, which has two domains, an exonuclease activity, as well as this N7 methyltransferase activity. For the N7 methyltransferase, we see that it has a zinc bound, as well as that cap protein binds in, and then it requires S-adenosylmethionine as a cofactor. Here we see they found it bound to SAH, which is the product after the methyl has been taken away. And so we have a nice structure here on which drug design can be developed. Our group here at Northwestern, in my research group, we solved the structure of NSP10 plus NSP16, which is this 2O methyltransferase that then adds it so it can be cloaked from the immune system. Here we see this very nice 1.8 angstrom structure with the S-adenosylmethionine bound into it. This is, again, that donor. We've also now solved this with the Cap0 bound into it, and that structure is still in refinement. So we're actually interested in our laboratory in looking at inhibitors of this particular protein now that we have the structure. We're working with groups that are doing computational, using our structure for computation to predict things that'll be novel inhibitors, inhibitors that might mimic the SAM molecule, as well as we're really potentially interested in peptide mimetics that will block the ability to NSP10 to bind. These are actually really of interest in part because NSP10 binding to NSP16 is essential for its activity. The NSP10 also has been shown in proteomic studies to actually bind to other things in our host cell that may be further developing the immune suppression. And so we think that if we can identify a peptide that affects this interface, it might also block interface of NSP10 with other immunosuppressive activities. And this might be very beneficial in terms of early treatment to enhance clearance of the virus before people become seriously ill. So there's a worldwide effort for structural biology for drug discovery. I've talked about our work in the Center for Structural Genomics of Infectious Diseases, as well as at our sister center at Seattle Structural Genomics of Infectious Diseases. Jihao Rao's lab at Tsinghua University in Beijing is incredibly active in this, and they've been active over the last 5 years and are really putting out nice work also, as well as work from the Diamond Light Source. What are the next steps? So we have lots of structures now. This is the earliest stages you can get in structure-based drug design. What we really want to know now is, can we repurpose FDA drugs? That requires development of assays, testing those, as well as looking at those ligands bound to these proteins in advanced crystallization studies. We want to retest lead compounds, either that were originally developed for SARS or that are coming, or new novel lead compounds coming from ligand screening or from fragment screening. We want to do continued structural biology of putative targets. We're actively working on NSP10 plus NSP14. And some of our structures are actually a little low resolution. For outstanding drug design, they really need to be less than 2 angstroms to really be able to see where all the side chains are positioned, and that work is also ongoing. From then, you refine those compounds with the chemist, you test lead compounds for antiviral activity, and then go into in vivo studies in pharmacokinetics. With regards to this next line, it is exciting that mouse models are starting to come online. Ralph Barrick's group in North Carolina has worked on SARS for years. They actually had developed a humanized ACE2 mouse model, but they had only a small colony effective for themselves. They have actually just this week presented their lead compound and have mouse studies to show that. They've shared these mice around the United States. Many different groups have actually developed their own transgenic mice based on their model. And so we expect that there'll be more mouse models for testing drugs coming online really now and in through the summer. And then the next step is really to move on into clinical trials. For repurposed drugs, those have already been through safety, so they can go straight into testing trials and into patients, which is what we see now happening with remdesivir. So we're off the starting blocks, and I think we're off to a great start. We've had record time. We've only learned of this virus on January 10th, and you see how much structural biology has already been achieved in less than 3 months. So gene to structure has been incredibly fast, and we want lead to drug to be just as fast. And with that, I'll just remind you that I'm speaking on behalf of a large consortium of investigators headquartered at Northwestern, but it's really been a fabulous group to work with, and I think we're making great progress. So I'm happy to take questions. Thank you.
All right, thank you, Dr. Satchell, for that presentation. We have some questions coming in. As a reminder to our audience, the Q&A app is c2st2.cnf.io. So we'll get started with our first one. There was a recent article in the New York Times stating that the genetic composition of the virus in New York City is more related to the virus found in Europe. What do we know about the differences between the strains that might be found in China versus the rest of Asia or Europe?
So right now, if you look at the phylogenetic tree, it looks like, I believe it's about 8 different branches. And so what genetics tells us is it's very easy from just minute changes, even a single nucleotide, to sort of tell how the virus has moved around the world. It's not clear yet whether any of these changes have impacted the function of the virus. We do know that there are places where we don't see mutations at all, which would tend to suggest that they're very essential. If they had gotten mutations, they led to unproductive infections that were never passed on. But these ones that we see persisting suggest that they're making virus just fine, and they're able to pass. So in Washington State, you know, we had 2 introductions, one originally from came twice in, and so you see 2 different branches there. We don't know a lot about what's going in Chicago. We don't have a lot of genomic data yet for Chicago. But from a drug discovery standpoint, we're looking at these now, and particularly through our bioinformatics group at UC Riverside, whether any of these mutations might have a strong impact on the drug discovery, trying to determine if we need to solve the structures of those to impact our drug discovery. We certainly don't want to make a drug that's going to work in New York, but not Washington State. And certainly we don't want to make a drug that's going to work in half the population in Chicago and not the other half.
Absolutely. All right. So our next question from the app is, how do scientists identify older drugs that may be repurposed for treatment for new diseases?
So the most effective way is to just, in an array ligand screening kind of way, you have an assay. That assay can be either biochemical, so that's what we can do with the proteases, is say, does it inhibit protease function? We have that is set up at the Purdue site as a high throughput screen, so they can screen through hundreds of molecules very quickly. Can also be done in viral testing. We're working with a group in Utah. They can actually, again, just screen. They add virus to cells and say what stops the virus from replicating, and that can also be done in a high throughput way.
Great. All right, so our next one is, can you talk a bit about the collaboration happening between researchers who are actively fighting COVID-19 and how this is helpful?
So there's a great interest. One of the most important things is the amount of sharing of information. For example, the ligand screen at Diamond was shared literally as they collected the data in 2 different streams. And so in the past, if someone did a fragment screen like that, they might keep it to themselves until they got a drug developed and had a lead compound. They may never share it at all because it would be so preparatory and useful. Instead, people are making it out there. We have deposited all of our structures literally from the point we finished refining them, made them publicly available. I didn't, you know, for 1016, it's a great drug target. It's been downloaded so many times. We've been contacted by groups around the world for information. They're doing computational work. We're doing it. My opinion is, you know, I hope somebody can use our structures to do good drug discovery.
It's great to hear that this really is a global effort. So how closely related do viruses have to be for treatments or vaccines to work on both?
So it depends on how the component is designed. So a vaccine can be very, very specific. We see this in the flu vaccine, where each flu vaccine is specific to the strain that's circulating that winter. That's why we have to be vaccinated every single year. We also see that prior exposure to SARS doesn't really lead to immunity to SARS-CoV-2. So a vaccine that would have been developed for SARS wouldn't have actually probably been applicable now. The same with drugs. If that drug is so specific for SARS-CoV-2, it may not be useful for something downstream. So it's very important to make sure that you're developing reagents to the more conserved part so that at least it's applicable to the SARS-CoV coronavirus family, but really beautifully would be all around the coronavirus tree. If we can develop something that could actually get to those that cause the common cold, we could do something for 1/3 of the cases of the common cold.
All right, that's great. We have time for a couple more questions here. The first is, while in a pandemic or other emergency situations, how much testing is completed before a drug or vaccine is available to the public?
I think that you still go through all the major steps. It's just that they go faster and possibly with more rapid assessment of the regulatory oversight. So you see the trial for remdesivir going forward, and really there's very little, they're all undergoing proper clinical research oversight, but they are being done, the process of getting that approval is being done much faster because people are putting in the effort and going through the process. But it still needs to be good research done properly, because we certainly don't want to bring something broadly applicable that is not known to be safe and is not efficacious.
Absolutely. All right, and this is going to be our final question with Dr. Satchell. What has been the most challenging for you and your team in developing potential treatments?
I think that the deep challenges have changed as we've gone. I think the delay in terms of gene to structure was getting the genes in, because everything had to be synthetic. And so there was just a 3-week delay where we weren't doing anything. I think the challenge now is moving into that next stage is, as we're getting the structures, and particularly as we get to ligands that might be effective, is really getting to the viral testing and the animal testing. These have to be done at BSL-3 level facilities by people who are skilled, and those people are going to be challenged to take on lots of different things coming from around the globe. Prioritizing what to test is going to become, I think, a major challenge.
Well, thank you. I want to take this time to thank Dr. Satchell for speaking with us today and being a part of our virtual series. Thank you, everybody, for tuning into today's program.